Assalamualaikum warahmatullahi wabarakatuh. Firstly, let me introduce myself. My name is Alia from Chemistry Department of Institut Teknologi 10 November Surabaya. Now, I want to present about my experiment with the title is the additions of different adsorbents in SA-PVA matrix for Bacillus subtilis immobilization on methylene blue decolorization. Introduction. The amount of industries in Indonesia, such as textile, food and beverage, cosmetic, and pharmaceutical, is higher. Their industrial processes have many advantages and disadvantages. One of the disadvantages that they are produced with water. A study reported that on coloring process, 10 until 15% dye was discharged in wastewater. Methylene blue or MB is one kind of synthetic dye that is cationic dye which could be harmful to human and animal and of course have negative effect for environment too. There are three methods for remediate dye wastewater. There are physical, chemical, and biological. But physical and chemical reported that it spend more cost than biological method. On biological method, we could use microbes such as fungi, bacteria, and microalgae. Immobilization technique is one kind of technique that use microbe. It has some benefit than free cell method. Immobilization technique usually using sodium alginate SA, which is biodegradable and non-toxic, and added with polyphenol alcohol PVA that could enhance the mechanical strength. But the addition of PVA could increase mass transfer resistance. So the additions of filler adsorbents such as bentonite, activated charcoal, kaolin, and conical powder is so important for the microbes. And for the microbe, we use Bacillus subtilis that have known as bacteria that could degrade dye wastewater, include MB. Materials and method. Here is the materials. Method, adsorbent preparations. So the adsorbent on the experiment were formed in powder. For bacteria culture preparations, stock Bacillus subtilis inoculated on nutrient agar first and then inoculated on Luria bertani broth. For bacteria immobilization, the culture of Bacillus subtilis was centrifuged to get the biomass and added with sterile water. After that, the biomass of bacteria mixed with SA-PVA hydrogels and the adsorbents, then dropped to calcium chloride to form beads. For MB biodecolorization process, 2 gram beads added into MB solution that have 100 milligram per liter concentrations and incubated at 35 degrees Celsius for 24 hours. The result was analyzed by using spectrophotometer UV-Vis. For beads that had the highest results on MB decolorization, we analyzed the effect of temperature and incubation time that could influence the beads. For effect of temperature, were incubated at 25 until 40 degrees Celsius for 24 hours, and for incubation time, were incubated for 6 until 30 hours at 35 degrees Celsius. Result and discussion. MB biodecolorations. To get the percentage of MB decolorations, was use this formula. From the calculation, we know that the beads without adsorbents additions had decolorization approximately 78.12%, and then the additions of activated charcoal to SA-PVA Bacillus subtilis bead had the highest result than other, that was 87.59%. Some studies reported that activated charcoal could absorb many dyes and has high surface area and micropores. For the effect of temperature, the ambient decolorations were increased for temperature 25 until 35 degrees Celsius and had optimum decolorations at 35 degrees Celsius, with the final concentration is 5.38 milligrams per liter. Bacillus subtilis had the optimum temperature to grow at 37 degrees Celsius. That's why on this experiment at 35 degrees Celsius had the highest result decolorations. And for the effect of incubation time, the decreasing concentrations was detected along with the increasing of incubation times from 6 until 24 hours. And Connie et al. Reported that on that time, enzyme activity on Bacillus subtilis saw the highest results. For the conclusions, the additions of different adsorbents such as choline, bentonite, activated charcoal, and conca powder in SA-PVA, Bacillus subtilis immobilized matrix on MB biodegradation was investigated. The additions of activated charcoal had the highest MB degradation among others, which is 87.59%. The gap of MB degradation by SA-PVA activated charcoal Bacillus subtilis against SA-PVA Bacillus subtilis is 9.47%. The optimum incubation temperature was reached at 35 degrees Celsius for 24 hours incubation times. That's all my presentations. Thank you.